First is the association. What is the association mapping and the QTL mapping? Association mapping sometimes we can call the GWAS. But GWAS is the important tool that is used for analysis of the association mapping to identify the significant SNP. Identify the significant SNP near the candidate gene that influence the particular trait. So, in the genomic association mapping, genome-wide association study of the GWAS tools used to identify the significant SNP that are present in the near candidate gene that affect the trait. Okay, by using the different model MLM, GLM, Blink, FarmCPU, and others model. Okay, and after the identification of the significant SNP, we identify the candidate gene. Candidate gene means the genes that present in the near the significant identified SNP that affect the probable that affect the trait. Okay, and in the association mapping, generally use the linkage disequilibrium, not using the linkage in this case the LD block use. And the association mapping most important is there, this association mapping, the historical recombination, this term should be remembered, historical recombination. Okay, historical recombination is used, and in the QTL mapping generally use the linkage, not the historical recombination. Linkage only recombination done by the artificial by human being, by artificially done the recombination that included in the QTL mapping. But in the association mapping, the diverse line used and diverse population used by landes, but diverse line use and the historical recombination included in the analysis of the LD size analysis, and after that we do the association mapping. Okay, in the QTL mapping, first we done the linkage map construction based on the recombination between two gene and between two different pair of the gene and or marker, and we done the linkage map of the each chromosome. Okay, after that we done that TET, and TET data analysis means in this case the TET and the marker that associated with each other, TET and marker associated with each other, then this marker used to identify the a QTL. Okay, and QTL that affect this particular TET. And after the QTL identification, then the validation of the QTL is most important. Maybe this is false positive because in different of the QTL model is there the simple interval mapping and simple and the composite interval mapping, multiple interval mapping. Okay, the most important use this composite interval mapping, and after that this, and I will discuss in another class the different the QTL mapping model which you used in the QTL mapping analysis. Okay, I will tell the association mapping today. So in association mapping, the difference between the association mapping you should know. What is the association mapping and the QTL mapping, the difference? Okay, most important, everyone's saying that what is the difference between association mapping and QTL mapping? Most of the people confuse that the association mapping and QTL mapping same, but it is actually not same. But it's association mapping taken the few information but few hypotheses from the QTL mapping. Okay, in association mapping, most important the difference is the recombination. First, I am told that recombination here use the historical recombination used, and here this in the not historical recombination not used. Okay, and second is the resolution. This is coming in exam. Resolution of association mapping is high or not. In association mapping, the resolution is high. I will describe why the resolution is high. And in the QTL mapping, the resolution is low. In the resolution is high mean that the confidence of interval less than 1 centimorgan in the association mapping, but in QTL mapping the the resolution and the say क्या बोलते हैं इसको confidence of interval is more less than 5, not say it is the less, because is the distance in the genomic region which identified and association having to identify the gene or candidate gene which present this region, because the recombination is more. So recombination window, most important term, the recombination window will be low in the association mapping. But here the QTL mapping, the recombination window is more. Recombination window is more, but is here. If the recombination window is low, so I and the present of the an identification क्या बोलते हैं resolution मान say critical क्या बोलते हैं critical windows will be low. Okay, and the next coming for the most important, the power. Okay, in association mapping, the power is association mapping is low, but here the QTL mapping, the power is high. Why the association mapping in the power is low and it is here is high? Then what? And first you should know the what is the power for the identification of the significant SNP or the marker at the QTL, or that identification of the power is the probability of identification of the genomic region associated with a particular trait. So it is a power means power is equal to probability of detection of a particular genomic region. Maybe it is candidate gene or QTL. Okay, maybe QTL or the candidate gene is the identification probability is the power. Here this association mapping, the power of QTL power is low in association mapping. Why? In association mapping the recombination is high. So if the gene or the QTL present here and recombination in high, then if the marker present here and the recombination window is here and this marker present a gene or QTL present here, this probability of the detection will be low, in association mapping. But if the marker present here, it can identify. But in the QTL mapping, the recombination window is more. This is present marker, same marker present here, but QTL marker present here. If the recombination window is more, means this chunk of genomic region and the marker and this QTL uh, segregate along with, okay, not segregate and transfer to the gamete as a chunk of DNA, okay, in as an association showing. If but this distance is more, so this probability will be high, because this detection of this gene or the QTL will be more, but this genomic region the probability will be more, but here it will be low. So in association mapping the marker should be more as compared to QTL mapping for increasing the power. So in association mapping the marker should be high as compared to QTL mapping. Okay, what is most important difference is theirs. Here's is the diverse line use, and here's generally biparental population use. Okay, biparental population. Next, the concept of LD. First, you should know the equilibrium. In equilibrium, the frequency of a particular genotype, if the A present P squares and the capital A small a present the 2PQ and the small a present the q square. You always know that. But if the disequilibrium is there, is disequilibrium there, when if the equilibrium is present, here the capital A, B, the probability of the frequency of the probability of the or the frequency of the AB will be is equal to probability of A, probability of B. Okay. Sorry, this is the frequency. This is the frequency of the A and B. Man AB gamete genotype will be equal to frequency of A and frequency of B. But if the this is equal to 0 means there is no disequilibrium. Also, you can say that there is the equilibrium present. Equilibrium present. Okay. If the difference is there, capital A with this gamete genotype frequency, uh, A into P is not 0, then you can say that there is the LD present. Okay. When disequilibrium present, not LD, disequilibrium present. Okay, not LD, only D, disequilibrium present. Either here, but LD when, but LD disequilibrium, you should know that term the disequilibrium when the linkage concept included, when the disequilibrium of the gamete or the present of the genotype or the frequency of the allele is difference is there due to linkage, then this is called the linkage disequilibrium. If the disequilibrium happen due to linkage, then this is called linkage disequilibrium. Okay. And linkage disequilibrium, you should remember, this is not purely linkage. Is linkage disequilibrium tell that the association association between okay, between true or non-linear, this term should be remember, non-linear association. Okay, non-linear association, this term should be there. Because if the if the chromosome there one chromosome and it is a two chromosome, this is the A is theirs and this is the B is theirs. In the under linkage condition, the linkage means the two allele but two gene should be present, the which are linked under link conditions should be there present in the single chromosome. But in the linkage disequilibrium, it is maybe present in the same chromosome or different chromosome, non-linear association. Okay, maybe present here but maybe present here. When any allele, any two allele uh, moved along with and to a single gamete and affect and associated with a particular trait, then this is called association mapping. Okay, that is called the linkage disequilibrium. Okay, and how to calculate the LD? Okay, first, this is already described that what is the meaning of the linkage disequilibrium. Okay, here's if the difference is 0 means here is the disequilibrium is 0 or this is present in the equilibrium condition. Okay, if the difference is theirs means the PAB uh, dot P small ab is not is equal to PA small b, P a small a capital B is not is equal to, then this is called disequilibrium. Many formula is there for the calculation of the D. First, I tell you that the three most important formula used for the estimation of the LD. First is the D, second is the D dash, and third is the R square. In R square is the most valuable for the association mapping studies, and generally this is used for the estimation of the linkage LDDK LDDK analysis, LDDK value analysis ক্ষেত্রে R square use करते हैं. Okay, and different formula of the three different formula is there. First is the this formula small ab minus P small b dot small a capital B, and second formula Second formula I already tell in the previous slide. That's P capital B is equal to capital A and frequency of capital B. This, and the third formula is there. Third formula is same, only capital A capital B. Here this small a small b, p small a small small b. Okay, this is the formula. Okay, and this is not suitable. First you should know the why the d square is not suitable, because you should remember d dash as d dash nay d is depends on depends on the frequency of allele. Okay, so this is not suitable for the in the linkage ld analysis. Okay, the most important is use the ld dash and the r square. This is the for ld dash formula is the, you should first the d dash formula in the next slide I just okay okay, first the d dash formula. Two formula is there. First is d ab by minimum p a small p b or p small a p capital B. Okay, this is the multiply. Which value will be low, that included in a denominator. If this value is low, then denominator use as a hairs. This should be removed. Okay, it is happen when d value is more than 0. This should be remembered, when the d value is more than 0. Okay, and here d then another formula is there, d dash d capital A capital B. Okay, capital A capital B minimum capital A P capital B B small a dot small b. The which value is will be low, that included in the denominator portion. Okay, and here's is this formula is used when the d value is less than 0. The one important term is there, d dash is not dependent on the frequency of allele. Okay, and the d dash value range from the 0 to 1, where the d value may be positive or negative. Our r square value range is also 0 to 1. And what is the formula for the calculation of the r square? R square sometime it is used for the linkage decanalysis. I will tell in the next slide. Okay, this is the r square. This is the formula is the dab square. This should be square here, not square used. And this formula capital S small a capital B small b. Okay, this formula used because it's square, always positive result will become, and this range from 0 to 1. Okay, and in r square value, I first you should know the what is the LDDK. What is the meaning of LDDK? Next slide I will describe. Okay, first you should know what is the mean the LDDK. If the true marker present here and recombination not coming here, then the LD block size will be more. If the recombination happening between these two marker, so LD block will be reduced. So this the reduction or the reduction of the LD block size due to recombination. This is called LD decay. LD decay. LD decay. If the recombination high, then LD decay will be high. Okay, an LD decay can be visualized by using this r square value. This is the r square value. R square value. There here the 0. It is a 2, 4, 6, 8. Sorry 0 point 0 point 0 point 4, 0.2, and the 1 maximum is 1. Here the LD block, how to degrade by LD decay, how is going on, can be visualized by using this plot. Here this LD block is reduced, decreased, if the physical distance will be more. And here's every point is described. Wait, this is the every point describe the ld between between two marker. Okay, any two marker, any point describe the ld between two marker. If the physical distance is increased, the ld block is decrease. Because why ld block is decrease but decrease happening? Because the if the marker hairs and the marker second marker position is increased, or the second marker position increase, or the physical distance increase, the recombination will be more. If the low physical distance is there, so the recombination will be less, so ld block size will be more. This is the representation of the ld decay by using the r square value. Okay, and is most important, r square is most suitable for the ld decay analysis. First, most important, this is the some term, and the some this is the coming in I think delta is coming in the previous SRF exam. First you should know the why this symbol used, and where this symbol used for the analysis of the LDDK. যেমন lambda used for the population genetic analysis. This should be remembered. Okay, this formula is not needed, this formula is not needed, up to this formula is needed, but you should know the what is the relationship between these symbols with for the ld analysis in different condition. যেমন delta, this is coming, delta is generally used for the epidemiology analysis. Okay, and the lambda used for the population genetic analysis. In case control studies, this q is used. Okay, and the most important, in plant generally use r square value. Okay, the concept of haplotype means a, a segment of genomic region, a segment of genomic region that stay together and generally without recombination. Generally, term should be remembered, generally no recombination, no recombine. Okay, but no recombination. And other time I have said that a segment, first you should know that first is a segment of DNA or the genome, DNA or the genome, or and that uh, transfer or move together, that move together are and generally showing no recombination, that is called haplotype. This segment is called haplotype. Haplotype also used for the analysis of the GWAS analysis. Okay, here you can say that in sequence by I can say the A C T A G C T. This is the haplotype. Okay, then here's A C T G G C T. Here's only one SNP change. This is called as a single nucleotide only change, and this transferred together, not recombined to each other. Okay, then this is called the haplotype. Is a haplotype 1, and this is called haplotype 2. Okay, Then why first important term, the why the SNP is biallelic. You should remember first condition, generally SNP is biallelic. I have seen the multi-allelic also, a multi-allelic SNP also during the analysis of the GWAS, but you should remember in the exam view, SNP is biallelic. First, you should know the why is SNP is biallelic. First, you should know the transition and transversion. You know, always you should know, you know the what is the transition and transversion. If the purine, uh, purine replaced by purine, okay, then this is called transition. Transition, you know, uh, A converted, A replaced by G means A replaced by G. When then this is called the transition. But opposite will also be there. G uh, G replaced by A. Then this is called transition. But and also the pyrimidine also, if the pyrimidine means C converted into T or T converted into C, this is called together transition. Transversion is means the purine is replaced by pyrimidine. Okay, then this is called the transversion. But in the mutation in the genomic DNA regulation, but DNA repairing system, or the any others formula which any others factor that regulate the or the affect the DNA, and that the transition is more because a generally J generally converted into G via ionosine, because deamination of the adenine converted into ionosine, ionosine converted into G. But adenine to direct T or C not possible. Okay, for this the transition is more. Similarly, C the deamination and methylation, 5-methylation is produce T. You know, 5-methyl of uracil is known as that thymine, means deamination is possible to produce the uracil. Uracil का methylation होगा, then it's produce the thymine. Then C to T is possible, but C to A or C to G is generally not possible. So then this is SNP is called by SNP called biallelic in nature. Biallelic in nature means A, T, C, G, A. This is present in the one DNA, and this is present in uh, this is okay. This this converted into this sequence converted into A, T, T, G, A, means here SNP is there. SNP, the others are same. Only here the C converted into T is possible. Okay, but this is called the biallelic nature. Biallelic means C one SNP and T is another SNP. If the A is theirs, then it is called triallelic. If the GA means for, if the multi-allelic concept is theirs, okay, multi-allelic concept is theirs, A C G A A T T G A, then here's the A is theirs, T A G A, and A T G G A. Here that you can find that four allele present. Each is SNP1, SNP2. When SNP2 nahi, SNP1 ka is the first allele of the SNP, and the second allele of this SNP, and this third allele and fourth allele. But it is not possible. Only it's possible in T. So generally it is called biallelic in nature. Okay. Then the population structure. What is the most important term is the population structure, important concept in the association mapping in the population structures. If you have population, okay, not related to each other, not related to each other. This is the ideal, this is called ideal population, where the each individual not related with the others. Okay, all are diverse in natures in the population, then called the ideal population. But generally found that, I should next slide I will describe this population structure. Okay, but in general practical condition in population, subpopulation formation is there. Some individual related to each other, some some lines related, highly related to each others. Here is a subpopulation 1, subpopulation 1. Okay, this is a subpopulation 2, and this is a subpopulation 3 or C, subpopulation C or the 3. Generally this is happen if the even the rice population, if the diverse population is collected and done the association mapping, and some are the come from the japonica, and some from the javanica, and some from the indica, then these three, the individual or the line coming from the japonica from a subpopulation here, the javanica, the individual who is coming in the taken in the population, that from the second population subpopulation, and here's the indica, the lines which coming from the indica, then from the third population subpopulation, and this is called the important term, the population stratification. Population stratification, this is from the population stratification that effect, that most effect on the association mapping. Okay, I will describe how the q matrix and the k matrix or the q population affect the association mapping in other slide. Okay, here the how to know the what individual coming for the which individual. I have tell the three population, this is the one population, this is the second population, and this is the third population. Okay, and it is a criteria that the individual, which individual, either if the 200 individual is theirs, and among them which individual present first population, which individual present in the second individual, second population, and third, we cannot identify it, but based on this graph we can say that the which individual associated for the particular population. Okay, it important criteria is there. If the genomic relation of the relation with this particular population is similar up to 0.8, up to 0.8, then this you should know that 0 point is the threshold value. If the individual, the genomic proportion is the similar with this particular population, then this is called and this can be included into this particular population. Similarly, this proportion and this individual can be included into theirs, and this, but here some admixture is theirs, here some admixture is theirs, some here's admixture is theirs, and here's. First you should know the what is the admixture. Admixtures means admixture. Okay, admixture means those individuals, the threshold value is less than 8, mean it the some proportion of the genomic related to a particular one population, and some portion of the genomic is related to with the uh, second population or the third population, then this admixture, this line, but this line is can be called as a admixture. Okay, you can separate based on the threshold value. Okay, this called population structure. You can this can be done, this can be developed by using the structure software in GWAS analysis. Structure softwares in GWAS analysis, it can be done, but the population structure can be identified by using the This is the Negbar joining T. Negbar joining T. This is done by the molecular marker, SNP marker based. I have done in my MSc work. Okay. Here my magic population four population is there. Four population is theirs. That also this is not done by me. This is not for my thesis. It is I have. My notes I have included here. Okay, here the four subpopulation. This is supported by others way. This is called PCA way population structure. Population structure done by the PCA principal component analysis. Here you can see that this is the population structure one. This is two. This is three, and this is four. And these are the admixture. These are the admixture. Okay, these are the admixture. Here you should know the Q matrix and the K matrix. The most important, sometime this question coming in SRF, what is the meaning of the Q matrix and what is the meaning of the K matrix? Q matrix mean Q is a matrix. Okay, here if you have the three population, means population 1, population 2, and population 3. And here's if you have the 200 SNP. Okay, 200 SNP, SNP 200 sorry, this is the individual. Sorry, this is individual, 200 individual, and this is the individual, this is the one individual. Okay, here the invert one, or the line, this line which included plant one, but genotype one, you can say the genotype one. This is called genotype 200. Okay, here the proportion of the this in the proportion of the particular population here, this is called this here, you can say that 0.9. Here the population is 0.5 and 0.5. 0.5, no, sorry, 0.05 and 0.05. Total should be 1. Total should be 1. Means the genome proportion of the genotype 1 is related or identical with the population 1 is 0.9, but 90% similar. Here the 5% similar, and this is the P3 is the 5% similar, and total should be 1. And that time this is called the Q matrix. Okay, this is called Q matrix. And what is the K matrix? But Kinsey matrix. Kinsey matrix is a square matrix, you can say. Okay. Here, sorry, in next, you can remove this. Okay, now. What is the K matrix? Here, number of genotype, column and row should be same. Number of genotype 1, genotype 2, genotype 200. Okay, here genotype will be 200, and genotype 1, genotype 2, and genotype 3. And here the genotype, the Q in K matrix will be 1, because these are the similar, and genome should be 100% similar. So this is the 1, and the and this is the if it 1, and maybe it will be 0.3 similar हो सकता है. It is should be, maybe it is 4 होता है. It is not is equal to 1, not is equal to 1, that not is equal to 1. Maybe 3 maybe 30% similar हो सकता है, 40% similar हो सकता है, maybe 0.05. If the genotype 4, maybe 5% similar भी हो सकता है. This is done by, and similarity checked by identical by descent. Generally identical by descent used to know how the genotype 1 and other genotype are related with each other through genomic proportion. Okay, what is the identical by descent? Identical by descent means the probability of a allele here's probably you can genotype 1 and genotype 2. Here's the zero point 0.3 and 0.3 means 30%. Okay, what does that mean? IBD mean genotype 1 A1 and sorry, ओके ओके ठीक है. ई आईबीडी मीन्स क्या है और आइडेंटिकल स्टेट मीन्स क्या है. इफ द इंडिविजुअल इफ द पेरेंट एक जीनोटाइप 1 प्रेजेंट ए वन एंड जीनोटाइप 2 प्रेजेंट A1 A2. ठीक है. Probability of this allele or this genotype this allele coming from a common and this allele coming from the same common ancestor if the coming, then this is called identical by descent. Identical descent means probability of the allele of a particular locus, means probability of a allele for a particular locus, for a particular locus coming from a common ancestor, then this is called identical by state. But this allele coming from A1 is the same allele, but this coming from the different parent or different background, then this is called identical by state. Identical, these are the sequence, nucleotide sequence, these are similar, but when this coming from the common ancestor, then this is called IBD. And when the coming from the different background, then this is the background 1 and background 2 in different population, then this is called. And then when there is no common ancestor, then this is called identical by state. So some are coming, the most important, the models which used in the GWAS. Most important model generally used, first is the GLM model. The models, okay, first models is GLM, general linear model. Then second is mixed linear model. Third is farm from CPU, okay, and third is blink model, okay. In first I discussed about the GLM. First, it is a common, okay, and its usefulness now is reduced, okay, is not used now for the GWAS analysis. These three are generally now not used in the GWAS analysis, but now this blink is used, okay. And what is the meaning of the GLM? In GLM, only Q matrix I have described as Q matrix and K matrix. Here's only Q matrix is used, not used, but K matrix not used. Here's, so if the Q matrix and K matrix both include the identification of the significance, if the both include the identification of the significance SNP, identification of the significant SNP will be means identification accuracy of identification will be high. So the false positive rate will be low if the Q matrix and K matrix included, okay. Because if the Q matrix is not included, there is a subpopulation is there, may lead to affect the association mapping. Here I can say the Q matrix is here included is good. Here's what is the effect of the population in the association mapping I have not discussed, but I can discuss shortly. If the subpopulation is hairs, subpopulation 1 hairs, and a allele is very high, it is a population 1, and this is the subpopulation. This subpopulation 1 is the subpopulation 2, and capital A allele is very low, very very low, but small a allele very high, maybe present, but this actually not associated with trait. But when the subpopulation of the Q matrix not included, here this maybe lead to the false positive rate, because if the subpopulation height is 100 centimeter and here the height is 60 centimeter, but here the A not associated with the height rate, but here the frequency is very high in the population 1, but here is low. It means that if when you run the GWAS analysis, it's showing that the A allele, A SNP associated with this height, but actually it is not associated with the height rate. But in the GLM it is good, the GLM include in the stratification is reduced, means is in considerate here the GWAS analysis, but K matrix should be included. K matrix should be included, because when K matrix mean, if the K matrix means if the I relationship between the relation identical, but a relationship between the two individual in the population, if the two individual genotype 1 and genotype 2 more identical, if the identical, then this mean that some chunk of genomic region transfer, because here no recombination will be there, and then showing that this chunk of genomic region, if this marker present here, and the state but candidate gene or the QTL present here, that's showing associated with this, but actually this is not associated. Here's maybe recombination is there's more here, okay. As for this reason, the K matrix should be included. Okay. But in the GLM, only the Q matrix used, but K matrix not used. So the for the not include the K matrix, the fall discovery rate will be high, but the and the identification of the MTA marketed association by significant SNP, identify significant SNP, will be more as compared to MLM, farm CPU, and Blink model. So the in the GLM model, that are a high number of the significant SNP identified. Next, I will discuss what is the meaning of the MLM. MLM. In MLM, Q matrix and K matrix both included, and K matrix, and you should know that what mixed linear model. In mixed linear model, both fixed effect, and random effect, random effect included. Okay, random effect included. In the mixed model, the fixed effect included Q matrix or the structure पॉपुलेशन स्ट्रक्चर एज ए फिक्स्ड इफेक्ट हिसाब से लेता है, इसके साथ लेता है मार्कर्स व्हिच यूज्ड इन द GWAS एनालिसिस और एसोसिएशन मैपिंग, दैट आल्सो इज बोथ यूज्ड एज ए फिक्स्ड इफेक्ट. ओके. इन random effect only K matrix use. Okay, Kinsey matrix used in the random effect. K matrix used as a random effect. Okay. Most important information is that in MLM, number of MTAs marketed association or the identification of the identified the significant SNP will be very low. Few MTAs identified in MLM as compared to GLM, because here the K matrix used. And here's most important term is coming in In MLM, the false negative, false false negative rate, negative rate is very high as compared to GLM. GLM as compared to GLM, but false negative not false discovery rate, but false positive, false positive rate is less than false positive less than GLM, but in GLM, false positive rate is very high as compared to others model. Okay. Okay. In MLM, why the false negative is more, and the why the number of identified MTS or the significant SNP low, you should know. Here's the stringency. Here the MLM, the first important term, the stringency. Stringency is very high because here schematics included, and most important term, the schematics won't include. And in MLM, and the kinship, the markers which used for both, kinematics analysis, kinematics formation plus association mapping. Here, so confounding effect. Same marker used for the kinematics, same marker used for the association mapping. For that reason, some the confounding effect is there in the MLM between the kinship and the genetic effect. The confounding effect is there. Kinship and the genetic effect, this confounding effect is there. For this reason, number of identified stringency is very high, so false negative rate is very high. For this reason, number of identified MTS very low. So the confounding effect and the use of the kinematics is increase the false negative rate, and false negative rate reduce the number of MTS. Okay. Then others two models are theirs. Other two models is theirs. Is the farm CPU models, farm CPU model, farm CPU model. This is also used, and the fourth is the blink model. Blink blink model. This is the Bayesian information linkage disequilibrium iteratively nested keyway. This is the Bayesian information linkage disequilibrium iteratively nested keyway. This is the formula. The formula means this is the explanation of the each word. And the blink, generally you should know the blink model generally now used for the GWAS analysis. And here's kinematics not use. Kinematics not use. Here the LD linkage disequilibrium use in blink models, and is reduce the false negative rate. False negative rate is reduced. Also, it's reduce the false discovery rate. False positive rates also reduce. False positive rate also reduce here. Blink. It is the optimum model that can be used in the GWAS. Okay. Here's the Bonferroni correction blink. You can use the Bonferroni correction for increase the stringency for identification of MTA. I will discuss about the Bonferroni correction in the next slide. Okay. Here, the in the previous years, in SRF exam, one question came here, that is a x-axis and y-axis denote which component. which component. X axis and y axis denote which component. You should know the x axis of the Manhattan plot. X axis of Manhattan plot denote the chromosomal position. The chromosomal position, okay? This is the x axis. This is called y axis. Here the x-axis denote the SNP, the chromosomal position, and each represent the significant each SNP. This is the SNP, okay? And y-axis represent the negative. Y-axis represent negative log p value. For each MTA marker state association in GWAS analysis, one p value is coming. If the negative log p value is taken, then it produce more than zero, and maybe 30, okay, up to 30, 40 maximum, and negative log p value. And generally, threshold value taken is the 3. Generally, in GWAS analysis, the negative log p value use threshold value is use the 3. If they use the 3 negative log p as a threshold value, number of identified SNP or MTA will be very high. Here, false discovery rate may be high. Maybe this not associated with a particular test. This is called SPAT. SPAT means chlorophyll content estimation. This is the SPAT meters, or SPAT chlorophyll content estimation for estimation. Okay, here maybe this is not associated with the SPAT tests or the chlorophyll content. This may be associated or not, but when the stringency increase, the threshold value increase of here that's 5.36. How to include this? How to optimize the threshold value? For this optimization, one important term is this Bonferroni correction. Bonferroni correction is needed. I will discuss with example what is the Bonferroni correction and how to calculate the Bonferroni correction in the next slide. When the Bonferroni correction is done, then it provides threshold value of the negative log p value. When use this threshold value in my GWAS analysis, my negative log p value is the 5.36. 5.36. If the threshold value is 5.36, only one single SNP is there. It is highly associated with the LDBI TET or the particular TET. Okay. And this is the quintal y plot. I think this is not coming in exam, but it may become coming in future, maybe come. You should know the quintal or the QQ y plot, or QQ plot. Here you should know this is the observed negative log p value. Observe negative log p value particular MTA. It is the expected negative log p value. If this showing the same means, and the regression line present, the growth value present in the same, then this is suitable. And some last a 3 SNP separated from the in the top portion means 1, 2, 3 SNP. These are the 3 associated with this particular test. Here only 1 SNP separated. Okay, then means observed value is more expected value. Then this is called, you can say that this is associated with your particular test. Okay. First, I will discuss what is the Bonferroni correction. I will take on a few minutes more. Okay, only 15 minutes. Okay. First, you should know what is the Bonferroni correction. Bonferroni correction. I already said that the Bonferroni correction why used for the stringency, for the increase the stringency increase करने के लिए Bonferroni correction करना पड़ता है, and reduce the false negative rate reduction करते, and to reduce the false positive rate, also false positive rate reduction के लिए use करता है. And what is the formula of the for the determining the what is the threshold value of the negative log p value. Okay, for the Bonferroni correction threshold value, you should by using formula you can determine uh, negative log negative log negative log by p value. P means probability. Probability 0.05. You can p value means you can say as significance. You how much you want to significant. You can use the your p value. If you more significance you will not need it. You can use the 0.001 or you can use the zero point 0.5. Generally 0.05 used. Okay, and here's the how it is a total now here the total number of snp, total number of snp used in the GWAS. I can write here uh, mm, balance Bonferroni corrected threshold value is negative log negative log. P means significance. P value is you can use the 0.05 and 0.001 or 0.001. Okay, and here if you use the total number of snp used in association mapping or the GWAS. Okay, in my GWAS analysis I use the p value 0.05 and the number of SNP which I have used the 11,574. That this is the and Bonferroni correction threshold value will be 5.36. For this you can say that this Bonferroni correction threshold value is 5.36. Okay, the what is the this is the most important. I will take only 10 minutes more. Okay, the what is the most important factor that affect the linkage disequilibrium and the association mapping. First, the most important factor is the mating pattern. Mating pattern. Okay, mating pattern means is a self pollinated or cross pollinated or random mating is theirs or not, or self pollination is theirs. If in how mating pattern regulate the linkage disequilibrium association mapping, I can say that if the mating pattern is a random mating is theirs in population, is random mating is theirs in population, it increase the recombination frequency will be very high if the random mating is there, because heterozygous condition will be high in random mating. And again here's again the recombination will be take place generation after generation. It is reduce the ld block size. You already described that if the recombination increase the ld block size will be narrow, so LD block will be degraded. If the recombination increase, the LD decay will be high and LD decay, LD size will be decreased. So mating, if the cell pollination is theirs, in random mating, the LD decrease. Okay, if cell pollination is theirs, it's maintain the homozygosity. And if the homozygosity a allele, this is the b allele and b allele. If the homozygosity is there, so there is no chance of recombination. But recombination is happening, it is double as like a double crossing over. Okay, in the cell pollination, here the recombination frequency is low, so LD size is high. If you should note that in association mapping, if in association mapping LD block if very high in high, then you should take only few SNP. Okay, if the LD size, decay is more and LD size is low, then you should use high amount of SNP bar marker. Okay, this is found in the cell pollinated plant, and LD low block size is found in the maize. So in maize, if you want to done the GWAS analysis, you should use the more SNP as compared to wheat or rice. Okay. Second factor, second factor is the second factor is the selection. Selection. Second factor is the selection. Okay. How selection affect the association mapping? How selection affect association mapping? You should know. If a plant you take a plant height. Okay, plant height. Okay, plant height, and it regulated by two gene, A and B gene, and this is present in one chromosome. Okay, this is present in one chromosome, this is present in second chromosome. Okay, if the both gene control a plant height, and the selection done for this this state for this plant height and plant height more, this is the favorable allele, and this is the favorable allele of the B gene. Okay, if the increase the plant height selected the only the long heighted plant. Okay, if the select the long heighted plant, it on include only AB genotype, but if you use the if you select the only the small heighted plants, small a small b, if you select it. Okay, because both affect the plant height. Okay, and A allele and B allele of the two genes affect favorably, then it present different chromosome but showing that these are associated with each other. If the select the plant height more, then two allele or gene present to different chromosome, but it's showing that this is the associated with this two gene. Okay, here's the you should know the selection increase the LD increase the LD, not LD block linkage distribution increase in selection. Okay, and it's okay. So this way, this association mapping effect. Third is third is population structure. I already described how the population structures subpopulation affect the association mapping and LD. Okay, fourth is the admixture. Admixture, I have used the admixture term, but it is the admixture here the this is a different. Here the admixture means the irregular transfer or transfer of gene from population 1 to population 2. Okay, population 2. If the a here the allele is A5 for a particular gene present, it is transferred to hairs. Okay, this is called admixture similar to migration. And due to admixture and the migration, the allelic frequency, this allele frequency, this is not normally before not present, but after the migration is present here, so it affect the frequency of different for this locus. Okay, frequency of allele for this locus. Okay, if the frequency of changes, then it affect the association mapping and linkage disequilibrium. Okay, LD effect. Next factor, most important, is genomic region. Five is the genomic region. How genomic region affect the association mapping? I already tell that if the this is the genome, and this portion is the recombination hotspot is theirs. This is the high recombination. High recombination is there. So here the LD decay will be more and LD block will be low. Okay, then that time the association may be more marker needed for this region. But if the other genomic region, the recombination is low, then the LD is more. Okay, this is happening if the high recombination found is the genomic genomic region that contain the gene region, but exonic region but genomic that region that contain the more gene. Okay, that produce the high recombination rate, but the heterochromatic region by repeated region, the DNA region contain the low recombination rate. So the LD will be more and LDDK will be low. Okay, here you can use the low SNP. Low SNP it is suitable. Okay, if no needed the more SNP. Okay, then the six is the kinship. I have already described the kinship matrix, how affect the association mapping and the confounding effect. I have already tell. Okay, the kinship matrix generally done by kinship generally done by the probability of the allele of a particular locus coming from a common ancestor by identical descent. But sometime identical state also be included. GWAS analysis. Okay, here and kinematic should be included the GWAS analysis by association mapping. Okay, and it is done by one software, Tassel software. First you shoot the Tassel software is theirs. Tassel. Okay, Tassel, TASSEL. Tassel software you use for the extraction of the k value. Okay, kinematic analysis, the Tassel can be used. Okay, and LD block you can develop by using also Tassel software. Okay, Tassel software and population structure with structure software, or the also you can use the ITOL software for the population structure analysis. And seven is the genetic depth. Genetic depth, you know the genetic depth and the bottleneck. Okay, both increase the LD linkage decay may both increase. Okay, you should remember the bottleneck and this and the genetic dip increase the LD size. Top 8 is the gene conversion. Gene conversions means if a allele is theirs, here's the small a allele is theirs. If the genomic some segment of transferred to same locus and it converted into a, then it increase the number of and frequency of the a allele in a population. If the frequency is changes in a population, that it affect the association mapping. You should remember the gene conversion. First important, the gene conversion tends to reduce the correlation between LD and the genetic distance between the loci closely related to each other. Okay, gene conversion reduce the LD. Okay, you should remember how which gene conversion reduce the LD. Selection increase the LD size. Bottleneck and the genetic depth increase the LD size. Okay, and the genomic region is depend on the if the क्या बोलते हैं सर पॉप mating pattern. Okay, sorry, mating pattern may be increase LD size, may be decrease based on the selfing is theirs and or the cross pollination is theirs. In mating pattern how affect, I want to tell some point. After that I will close for the today's live class. Okay, how the mating pattern affect. One formula is theirs, you should remember for the mathematical point of view in the exam. Okay, Mating pattern. Mating pattern. Here's one formula is theirs. Crossing over rate, cross over a rate is equal to 1 minus s, 2 minus s. Okay. Here this is the helping percentage. Helping percentage. If the population is a self pollinated plant, here's will be s will be 100. 100 means 1. Okay. And 2 minus 1 means 1 minus 1 minus 1 means 0, means crossing over recombination rate is low. Okay. If the cell pollination is 100%, if the cell pollination 100%, if the cell pollination rate is 95%, if the helping is 95%, then what will be the crossing over rate? If the crossing over rate is reduced, the ld block will be high. If the percentage of the 95, then it will be 0.95 by 2.95. Okay. 1 minus 0.95 by 1.05 by 1 by 95 1.105. Okay. It affect, but it's reduced. It's I think it's reduced the less than 1. It is a less than 1. Okay. Less than 1. So crossing over rate will be low. Crossing over rate will be more. If it is a cost with the 100, if the 100 is cost, if the 0.9 है पकड़ लो, इसका value आया है, इसका percentage if you call, if it is cost with the 100, then it's produce the 9%. 0 point I think इसका 10% less than 10% होना चाहिए. ठीक है. I think is 0.09. 0. Okay, 0.09. Can you calculate? Wait. 95 by 105. It is coming here 1 minus 0.90. So minus 1 means 0.90. This is coming 0.95. Okay. Then if you multiply it with 100, then it produce 9.5 percentage of crossing over. You should remember, if the selfing is more than 95, more than selfing percentage is more than 95, the crossing over rate will be less than 10 percentage. You should remember, if the crossing over selfing rate is 95%, then the crossing over rate will be less than 2%. If the crossing over rate is decreased, ld block will be more, and markers needed for the association mapping will be low.